The brain, a network of inhibitory and excitatory synapses numbering in the billions that function as one to control thought, memory, motor control, etc. Imbalances between excitatory and inhibitory synapses are associated with diseases such as autism spectrum disorders and schizophrenia. This video will discuss the synaptic adhesion molecule, Neurexin, a type 1 membrane protein that spans the synapse. We will explain how alternative splicing and mutations of the protein lead to small structural changes which modulate synapse formation. Neurodevelopmental disorders are tied up with these small structural changes, making the study of Neurexin alternative splicing a promising area of research. Neurexins have 5 alternative splice sites. At each site, a particular exon is either spliced in or out of the mature mRNA transcript, eventually leading to a protein with a slightly different structure, called an isoform. Neurexin interacts with a postsynaptic ligand called neuroligin at an interface within the synapse. The interface is mediated by an octahedral coordination of calcium by 2 water molecules and 4 oxygens from the Neurexin. Salt bridges between Neurexin and neuroligin strengthen this interface. Interface strength is affected by splicing. Here are 2 examples of how this occurs in one type of Neurexin. 1. Splice site B encodes for an insert which disrupts the position of glutamate, making it unable to form a salt bridge to neuroligin. 2. Splice site 4 encodes for an insert which forms the S-alpha-1 helix. This helix forces a glutamine into a 310 helix such that it too can coordinate the calcium ion. Because structural changes such as these affect binding affinity, the balance of excitatory versus inhibitory synapses may change. Let's see how. Specific isoforms of neuroligin are found at either inhibitory or excitatory synapses. Neuroligin 1 tends to appear at excitatory synapses. Neurexin 1 beta can only form a stable interaction with this neuroligin if it does not have an insert at splice site 4. Changing the splicing of a neurexin will change the delicate balance between excitatory and inhibitory synapses, perhaps leading to a neurodevelopmental disorder. In addition to the effects of splicing on disorders, the role of mutations has been studied as well. Mutations in these proteins have also been linked with the aforementioned disorders. Early studies found that autism is heavily linked with improper signaling in the brain. Because neurexins and neuroligands are involved in signaling, mutations in these proteins have been studied in relation to neurodevelopmental disorders. Specifically, point mutations in the genes for neuroligin 3 and neuroligin 4 have been linked to abnormal neurodevelopmental and neuropsychiatric outcomes. The 2 mutations that were found were a nonsense mutation in neuroligin 4 and an aspartate, and a missense mutation in neuroligin 3, changing arginine to cysteine. As the amino acid sequences for both these proteins are very similar, the effects of these mutations can be compared to the same wild-type sequence. The wild type of both neuroligin proteins has a molecular weight of about 130 kilodaltons. The point mutation in neuroligin 3 produces a protein of correct length, just with a cysteine replacing its typical arginine. On the other hand, the nonsense mutation in neuroligin 4 results in a truncated protein of only 60 kilodaltons. Both of these mutations impair synapse formation due to intracellular retention of neuroligin in the ER, preventing proper function of the synapse. The neuroligin 3 mutant, while only a missense mutation, was found to decrease surface levels of protein by 4 to 5-fold in the mutated cell. The neuroligin 4 mutant is absent from the cell surface entirely. Due to neuroligin inaccessibility, an interface with neurexin cannot form, inhibiting synaptic efficacy. The deleterious effects of these mutations show how neurexins and neuroligins are crucial for proper synaptic development. Neurexin 1 deletions have also been implicated in a large range of developmental disorders. In a cohort of 21 children with Neurexin 1 deletions, ranging from 74 to 702 kilobases, 69% had speech disorders and 57% had difficulty with receptive and/or expressive language. Similarly, in another study of over 3,000 individuals, the phenotypes of those with Neurexin 1 deletions included autism spectrum disorders, mental retardation, language delays, and hypotonia. These data suggest that mutations of Neurexin 1 impair the proper development of cognitive function. As the national prevalence of neurodevelopmental disorders has increased more than 17% in the past 20 years, the research into the origins of these conditions is crucial to furthering medicine, developing treatment, and improving quality of life. Using modern-day techniques of gene editing, such as CRISPR-Cas9, we may soon be able to reduce the risk of neurodevelopmental disorders or provide treatments for existing conditions. To use CRISPR-Cas9, we need to briefly understand how splicing occurs. The protein p66-alpha binds to neurexin genes and forms a complex that methylates the DNA. In the case of some neurexins, this methylation is required for the exon to be spliced in. We can imagine using this knowledge to develop techniques for potentially preventing neurodevelopmental disorders. If we know a certain disorder is due to excess neural excitation, and we know a particular exon splice in Neurexin leads to excitatory synapse formation, a potential preventative measure would be to prevent inclusion of that exon. Using CRISPR-Cas9, we could imagine mutating the binding site for p66-alpha such that the methylation complex does not form, the exon is not spliced in, and excitatory synapses do not form. This is simply one idea of where Neurexin research might go.